In our last class, we have discussed about the reproduction in organisms, that is asexual and sexual. We have discussed the types of asexual reproduction and discussed the details about the binary fission and the multiple fission. Now today we are going to begin our lesson with the next type of asexual reproduction, that is known as budding. Now here the hydra is shown for the budding. The yeast also undergoes the process of budding. Now in case of budding, a small bud develops on the parent body and that keeps on growing in size. This bud can remain attached to the parent body or it can get detached also. But here also the scheme for the division is karyokinesis is followed by cytokinesis. That is, the nucleus divide first and then after the division of the nucleus, the cytoplasmic division occurs. The next method is the method that is known as fragmentation. Now in the fragmentation, the body of the organism is broken into pieces or fragments and each piece or a fragment develops into new individual. Here the planaria is shown, that is flatworm, and the body of the planaria is broken into three pieces, that is head, middle piece and tail, and you can see that every piece had developed into new planaria. So this is fragmentation. Planaria also shows regeneration. Now in the regeneration, the lower grade of organisms regenerate the whole body. The way here the planaria is regenerating the complete body. But as you talk about the regeneration in the higher groups or the higher grade of organization, the regeneration capacity decreases. The starfish can regenerate its lost arm. The lizard can regenerate its lost tail. We also show the process of regeneration, but we can only regenerate our dead parts like nails, hairs, skin. So from this discussion it is very clear that as the grade of organization increases, the regeneration capacity of the organism decreases. The next method of asexual reproduction is spore formation. The spore formation is generally seen in fungi and bacteria. The spores are generally found in unfavorable condition. Rhizopus is the common example. It is commonly known as a bread mold. You all have observed a green blue color fungi that develops on the bread when it gets moistened up. So this is the same fungi and the spores are responsible for development of that fungi. Now you can see the fungi body, which is labeled as a hyphae. On the hyphae is a round ball like structure which is known as sporangia, and within the sporangia the spores are produced. Till the time the conditions are unfavorable, the spores are present within the sporangia, and as the condition become favorable, the spores are dispersed, and these spores when come in contact with moisture, humidity, temperature, they sprout out and give rise to new individual. Now the next method for the propagation is the vegetative propagation. Now you have already known this method. Vegetative propagation means when the part of the plant like leaf, stem or root is used for obtaining a new plant. Vegetative propagation is further divided into two types, that is natural vegetative propagation, which occurs in nature on its own, and artificial vegetative propagation, for which the human beings are required. So for artificial, we will see the first method, that is cutting. Now in cutting, the stem cutting or root cutting is taken and that is sown in another pot, and from that cutted part a new plant arises. You must have visited several times at your friend's home and seen the very beautiful rose flowers. So after seeing them, what you do? You bring the stem cutting from their home and then you put that stem cutting in another pot for obtaining the new rose plant at your home. So this is the process that is known as cutting. The next method is the grafting. Now in the grafting method, the vascular tissue of the plant is mixed. The two plants are taken with the different characteristics, and the one plant is in potted condition, which is known as stalk. The other plant, which is in unpotted condition, is known as scion. The vascular tissues are merged for the stalk and scion, and then they are covered with the moist soil and further wrapped with the butter paper or the cellophane paper so that the exchange of gases can occur. Mango, strawberry, then rose are propagated by the method that is known as grafting. The last method for the artificial vegetative propagation is the layering. Now in this layering, the condition applied is that the stem of the plant should be soft or flexible, because you need to bend the stem and then you need to bury the stem in the soil. The part of the stem that you bury in the soil develops the root, and from that part another plant arises. You can see the figure. This method is generally used for strawberry, guava, because they have got the flexible stems. This is all about the methods that are involved in asexual reproduction. Vegetative propagation is very advantageous in a way that through this we can propagate or we can obtain a new plant very easily. The seeds, the flowers and the fruits can be obtained in less time or a duration. Then the seedless plants or the plants which do not produce viable seeds, they can be propagated very easily by this method, like banana, grapes. They can be propagated easily by this method. The way it had got advantages, the disadvantages are also there. That is, with the positive characters, the negative characters also go to next generation. Then no new variation or no new adaptation is found in the plant, because this method is the asexual method, where the parent give rise to new individual, which is the Xerox copy, that is the clone. So these are the advantages and the disadvantages of the method that is vegetative propagation. This is all for the day today. Thank you.